Hello, everyone, and welcome. Today, we're going to dive into a really fascinating topic, the sexual reproduction of flowering plants. Now, I know this is a super important chapter for your exams, especially for Neet, but it's also just an amazing story, right? It's about how the beautiful flowers you see all around you actually create the seeds for the next generation. So let's decode it together, step by step, straight from your NCERT textbook. You know, the textbook kicks things off with a really powerful thought. I mean, think about it. When you see a beautiful flower with its amazing colors and that incredible scent, what do you think? It's easy to assume it's all there for us to enjoy, right? But the biological reality is so much cooler. Every single one of those features, the color, the perfume, everything, is actually a highly specialized tool. And it has one job, one crucial mission, to help with sexual reproduction. Okay, but before we jump into the nitty-gritty, it's really important to take a moment and acknowledge a true pioneer, Professor Panchanama Maheshwari. This man was, well, a giant in the field of plant embryology. I'm talking about the science of how plant embryos actually develop. His incredible work was foundational in creating the very first NCERT biology textbooks back in 1964. So as we go through things like the embryo sac and double fertilization today, just remember, we're standing on the shoulders of the great work he did. So here's our game plan for today. First, we're going to look at the key players, the male and female parts of the flower. Then, we'll see how they meet up through pollination. After that, we'll dive into the main event, fertilization. And finally, we'll check out the results of all that hard work, how seeds and fruits are formed. Sounds good? All right, let's get started. Okay, let's kick things off with our very first section. We'll begin with the flower itself, which your textbook rightly calls a fascinating organ, and it really is. From there, we're going to zoom right in on the male reproductive structures. We'll follow the entire journey, starting from the stamen and going all the way down to the tiny but incredibly important pollen grain. So take a look at this. What you're seeing here is a longitudinal section, a slice right down the middle of a typical flower. Now, you probably recognize the outer parts, right? The sepals and the colorful petals. But the real action is happening right here, at the very heart of the flower. This is where the reproductive parts are. We're going to be focusing on two main structures. The Androecium, that's just the fancy name for all the male parts, the stamens, and the Gynoecium, which is the female part, also called the pistil. Okay, let's zoom in even closer on the stamen. This is the individual male unit. You can see it's made of two main parts. There's this long, slender stalk called the filament, and its job is to hold up the anther. Now, the anther, that's the business end. It's usually bilobed, which just means it has two lobes. And each of those lobes is diathecous, another term that simply means it has two chambers. So if you do the math, that gives the anther a four-sided or tetragonal structure. And inside that structure, you'll find four very important pockets called microsporangia. And this is exactly where all the pollen gets made. All right, now let's peek inside one of these microsporangia. You'll see it's really well protected by four distinct wall layers. The outer three, the epidermis, endothecium, and the middle layers, you can think of them like a protective shell. They not only guard what's inside, but they also help the anther to split open, a process called dehiscence, to let the pollen out. But it's this innermost layer, the tapetum, that's really special. Its job is to nourish the developing pollen grains. It's like a nursery providing all the food they need. And right at the center of all this, you have the sporogenous tissue. This is the starting material, the stuff that will become pollen. So you might be asking, how do we get from that sporogenous tissue to actual pollen grains? Well, here's how it works. Each one of those cells can become what's called a pollen mother cell, or PMC for short. Now pay close attention here. This is key. The PMC is diploid, meaning it has a full set of chromosomes. It then goes through meiosis. Remember that? The special cell division that cuts the chromosome number in half? This whole process is called microsporogenesis, and the result is a little cluster of four haploid cells, known as a microspore tetrad. This is the absolutely crucial first step in making the male gametes. Now, as these microspores start to mature, they develop a super tough, two-layered wall. The outer layer is called the exene. Seriously, think of this as a suit of armor. It's made of a substance called sporopollenin, which is one of the most resistant organic materials known to science. It can withstand high temperatures, strong acids, you name it. It's so tough, in fact, that it allows pollen to be preserved as fossils for millions of years. Now, this armor isn't completely sealed. It has these little openings called germ pores, which are like tiny escape hatches for later. And then, inside that, you have the inner wall, the intine. It's a much softer, continuous layer made of cellulose and pectin. So what's actually inside a mature pollen grain when it's ready to go? Well, it is the male gametophyte, and it has two cells inside. You've got the big one, the vegetative cell. Think of this as the packed lunch for the journey. It's full of food reserves. And then, kind of floating inside the vegetative cell, you have the smaller generative cell. And this is the important one, because this is the cell that's going to form the male gametes. And here's a cool little detail. In about 60% of flowering plants, the pollen gets shed at this two-celled stage. But in the other 40%, that generative cell divides one more time before the pollen leaves the anther, so it's actually shed at a three-celled stage. All right, so we've got the male side of the story covered. Now, let's switch gears and look at the female reproductive structure, the gynoecium. We're going to examine the pistil, then take a deep dive inside to the ovule, and finally, we'll see exactly how the female gametophyte, which we call the embryo sac, is formed. So the gynoecium is the entire female part of the flower, and its individual units are called pistils, or sometimes carpels. It's pretty easy to spot its three main parts. Right at the top, you have the stigma. This is basically the landing pad for pollen. It's often sticky to help it catch. Then you have the long, stalk-like structure called the style, and that connects down to the swollen base, which is the ovary. It's inside the ovary that you'll find the ovules, and these are the little structures that have the potential to become seeds. Okay, let's break down the ovule itself. It's also known as the megasporangium. A good way to think of it is like a tiny protected fortress. It's attached to the ovary by a little stalk called the funicle. The main body of the fortress is surrounded by protective layers called integuments. Now, these walls don't seal up completely. They leave a tiny little opening, a gateway, called the micropyle, and this is where the pollen tube will eventually enter. Inside all these protective layers, you have the nucleus, which is a mass of tissue packed with food reserves. And right in the very center, at the heart of it all, is the main event, the embryo sac. This is the female gametophyte. The way this female gametophyte develops is really fascinating. It starts with just one special cell in the nucellus, the megaspore mother cell, or MMC. Just like on the male side, this diploid cell undergoes meiosis, and it produces 4 haploid megaspores. But here's where things get different. On the male side, all 4 survived. Here, 3 of them just degenerate and die. Only 1 survives. This single surviving cell is called bifunctional megaspore. From this one cell, the entire embryo sac will develop. Its nucleus then divides 3 times in a row, but through mitosis, not meiosis, so the chromosome number stays the same. 1 nucleus becomes 2, 2 become 4, and 4 become 8. After you have these 8 nuclei, cell walls finally form, organizing them into that very specific 7-celled 8-nucleate structure. And here it is, the final structure of the mature embryo sac. Now, you'll always hear it described as 7-celled and 8-nucleate. It sounds a bit confusing, but let's break it down. It's actually very organized. At the end, near the micropyle opening, you have what's called the egg apparatus. This is made of 1 egg cell and 2 helper cells on either side called synergids. At the complete opposite end, you have 3 cells called antipodals. So that's 6 cells so far. The 7th cell is the massive central cell that takes up most of the space. But this big cell has 2 nuclei inside it, the polar nuclei. So there you go, 7 cells, 8 nuclei, and it is now perfectly ready for fertilization. So let's recap. We've got the male gametes all packed up inside the pollen grain. We've got the female gamete waiting inside the embryo sac. But there's a problem, right? Neither of them can move on their own. They're non-motile. So how do they get together? Well, that's where pollination comes in. Pollination is simply the process of transferring the pollen grains from the anther over to the stigma of a pistil. Okay, depending on where the pollen comes from, we can classify pollination into 3 types. First, you have autogamy. That's pollination within the same flower. Super simple. Then you have geitonogamy. Now, this one's a little tricky. This is when pollen from one flower lands on the stigma of another flower, but on the very same plant. So functionally, it looks like cross-pollination because a pollinator, like a bee, is usually involved. But genetically, it's self-pollination because it's the same plant. Finally, we have xenogamy. This is the only type that is true cross-pollination, where pollen from a flower on one plant is transferred to a flower on a completely different plant. This is what brings genetic variation. So how does pollen actually travel? It needs help from external agents. These agents can be abiotic, meaning non-living, like wind and water. Now, this is really a game of chance, isn't it? So these plants don't waste energy making pretty flowers. Instead, they produce enormous amounts of pollen that's super light and non-sticky, just hoping the wind carries some of it to the right spot. The other strategy is to use biotic agents, living things like insects, birds, bats. These plants go all out on advertising. They have large, colorful, fragrant flowers, and they offer rewards like nectar, all to attract their animal partners to do the job much more efficiently. Now, even though self-pollination seems easy, continuous self-pollination can cause something called inbreeding depression, which reduces the fitness of the offspring. So to avoid this and encourage cross-pollination, plants have evolved some really clever tricks, or outbreeding devices. For example, some plants have a timing issue. The pollen is released before the stigma is ready or vice versa. In others, it's about position. The anther and stigma are placed so far apart that self-pollination is unlikely. Some have a genetic lock and key system called self-incompatibility, where the pistil can recognize and reject its own pollen. And, of course, the most straightforward method is to produce unisexual flowers, which makes self-pollination impossible from the start.
All right, so the gametes are ready and pollination has happened. We've now reached the climax of our story. Fertilization, and all the incredible events that happen after. And it all kicks off with a really important conversation, a chemical dialogue between the pollen grain and the pistil. You see, just because a pollen grain lands on a stigma doesn't mean fertilization is a done deal. The pistil has to recognize if the pollen is the right type, if it's compatible. This whole interaction is a dynamic process, a chemical conversation. If the pollen is of the right type, the pistil accepts it. The pollen grain then germinates, growing a long tube, the pollen tube, down through the style towards the ovule. But if the pollen is the wrong type, incompatible, the pistil rejects it, and the pollen tube simply won't grow. And now we come to the main event, a process that is completely unique to flowering plants. It's called double fertilization. So after the pollen tube enters the ovule and reaches the embryo sac, it releases 2 male gametes. And here's where the double part comes in. The first fusion event is called syngamy. One male gamete fuses with the egg cell, and this forms the diploid zygote, the cell that will grow into the embryo. At the same time, the second fusion event happens. This is called triple fusion. The other male gamete fuses with the 2 polar nuclei in that big central cell. So you have 3 nuclei fusing together, which is why it's called triple fusion. This creates a triploid cell called the primary endosperm nucleus, or PEN, 2 fertilizations happening in one go. It's absolutely remarkable and a hallmark of all angiosperms. So what happens after this incredible event of double fertilization? Well, development kicks off. First, that primary endosperm nucleus develops into the endosperm. The endosperm's job is to be the food supply for the embryo. It's like making the baby food before the baby starts to grow. Only after the endosperm is established does the zygote start developing into the embryo. The best example for endosperm is a coconut. The coconut water you drink, that's free nuclear endosperm, and the white kernel, the part you eat, that's the cellular endosperm. So as all of this happens, the ovule matures into a seed, and the ovary develops into the fruit that surrounds it. So let's wrap up with a question for you to think about, one that connects a lot of these ideas. We know that a typical dicot embryo has 2 cotyledons, and a monocot embryo has just one, which is called the scutellum. So my question is this: how do you think this fundamental difference in structure relates to how each plant stores food for its baby embryo? Think about it. It's a fantastic example of that classic biological principle. Structure is related to function. We'll touch on that and much more in our next session. Thanks so much for joining me today.